आज आपका टॉपिक है जो अभी हमने कंप्लीट नहीं किया है ह्यूमन जीनोम प्रोजेक्ट. It is a project used to identify the human genome, to know the base pairs present in the human genome, and it took about 13 years to complete the project. As the project was started in 1990 and it got completed in 2003. That means it took 13 years to complete the project. It was organized by the US Department of Energy. US Department of Energy, and also it was organized by National Institute of Health, National Institute of Health, Wellcome Trust of US also joined the organization. Wellcome Trust of US also joined the organization, and it is the Human Genome Project which helped us in the diagnosis, treatment, and prevention of so many genetic disorders. Prevention of so many genetic disorders. So many countries took part in the formulation of the project, like it was Japan, it was France, Germany, China, and UK, etc., United Kingdom. This project now used for the diagnosis, treatment, and prevention of the genetic disorders, in addition to the identification of the base pairs. This human genome project was also applied for non-humans. Non-humans पर भी human genome project apply किया गया है in order to know the genome of some other organisms which give us some useful products, like on bacteria, on yeast, bacteria E. coli, yeast, Saccharomyces cerevisiae, and also a nematode, Caenorhabditis elegans. Caenorhabditis elegans. It is a non-parasitic nematode which is not pathogenic, non-pathogenic nematode, Caenorhabditis elegans, which gives us products for human use. There are so many bacteria which are useful for human use, which give us products having desirable genes. They give various medicines, various products, and also yeast is used to obtain desirable material for the human welfare. So, Human Genome Project was also applied on these non-humans like bacteria, yeast, and so many others like Caenorhabditis elegans, which is a non-pathogenic nematode. Also on Drosophila, it was applied. Drosophila means fruit fly. It was also applied on some plants like rice. Rice plant पर भी ये Human Genome Project apply किया गया है. And also on another plant like Arabidopsis. Arabidopsis is the plant on which HGP has been applied. Now, methodologies. There are two methodologies. There are two methods used for the formulation of the Human Genome Project. Like, there is the first approach to identify all the genes. We have to identify the genes in the human genome which are expressed as RNA molecules. RNA molecule वाले genes हमें अलग करने हैं, which is known as expressed sequence tags, ESTs. Expressed sequence tags. It means to identify the genes which are expressed in the genome as RNA molecules, expressed sequence tags. There is another approach, that is sequencing the remaining genome. Then sequencing the remaining genome. Remaining genome means both coding and non-coding regions. Coding regions be, non-coding regions be. Sequencing the genome, both coding and non-coding, it is known as sequence annotation. Sequence annotation. Sequence annotation. Now, the sequencing is done from both ends. From both ends, sequencing is done. For sequencing the human genome, there is a technique developed by Frederick, which is known as sequence annotation. Sequence annotation, it is developed by Frederick. The sequencing of human genome has been developed by Frederick. Now sequencing is done from both ends. First of all, the DNA is isolated from the cell. Isolation of the DNA is done first. Isolation of the DNA. When DNA is isolated, it is cut into fragments. Fragments are cut from the DNA. It is so long, it cannot be sequenced wholly. It can be cut first into fragments. And these fragments are later on cloned. Cloning of the fragments means amplification in order to get large number of desired fragments. Amplification. After amplification of these fragments, cloning of these fragments, then sequencing is started from both the ends. The fragments are cloned in a host cell using vectors. Host cell means we can use bacteria, we can use yeast, we can use some another cell in which the desired fragment is introduced for the purpose of multiplication, for the purpose of cloning. And special suitable vectors are used. Sometimes bacteria are used, which is known as BAC, bacterial artificial chromosome. Sometimes yeasts are used as vectors, which is known as yeast artificial chromosome. And later on, these fragments are cloned. They are cloned, and later on they are sequenced. Sequencing is done from both the ends. It is known as automated DNA sequencing. Automated DNA sequencing. Automated DNA sequencing. बाकी हम tomorrow